You're listening to Conferences Online Allergy from Children's Mercy Hospital in Kansas City, Missouri. Today is September 6, 2024. Our topic today is nuts and bolts of genetic testing. This is presented by Dr. Bonnie Sullivan. She is a clinical geneticist and assistant professor of pediatrics at Children's Mercy Hospital in Kansas City, Missouri.
So today we're kind of going to go through a big overhaul of nuts and bolts of genetic testing. We're going to talk about a ton of different tests. We're going to talk about a ton of different technologies so that you guys feel more comfortable as it becomes more and more a part of clinical practice. From a disclosure standpoint, I sadly don't have any, so nothing exciting there. And then from the objectives, we're going to review basics of DNA. And I know you're probably like, oh, not that. And we'll go fast, but I also think it's important to have that foundation, and sometimes it's just nice to have definitions in one place. If you don't do this all the time, sometimes you forget about these things and it's assumed that you should know these things, but they slip your mind, and that's okay. And so I want to make sure you've got foundation so that you understand what you're doing when you're ordering testing and what you're looking at. We're going to discuss a ton of different types of genetic testing. We'll talk about limitations. We'll talk about logistics. We'll also review some updates in general, kind of specific to you guys as you are in training to become stellar allergist immunologists, what the research is showing and how genetic testing is being used. So to get us started, let's kind of go back to basic science and walk through what we're actually looking at. So what is DNA? I think everybody knows this, right? It's that double helix. It's made up of 4 different bases. There's adenine, cytosine, guanine, and thymine. And there's that sugar phosphate backbone that's helping link everything together so that we have this structure. And those ACGT are kind of the letters of our genetic code. That's how I think about them. And so as we continue going on, we'll talk about those letters being spell checked, because that's really essentially what a lot of our technology is doing these days. But this is kind of the bare bones basics of what we're looking at. When DNA gets read, it's read in these groups of 3, and that's called a codon. And each codon actually codes for that specific amino acid. And it's either the amino acid or specifically like a stop codon or the start codon. And so this is just the picture of all of the different types of codes to get you to each of those amino acids. Okay, I recognize these are probably things you are very comfortable with, but it's our starting point to make sure that we're all on the same page. Now, this is that central dogma that I swear we started learning in high school biology, right? Where we have DNA and it'll replicate itself, so we have lots of copies. And then we have transcription, which is the process of making the RNA. And then the RNA undergoes translation so that we get that protein product. And the idea from that is that, well, gosh, if we have a change in our DNA, we're not making the appropriate protein, and that could have all sorts of consequences for the body. But this is just that general principle there. A few definitions: genes. Those are a stretch of DNA sequence that are needed to make a functional product. You'll hear us talk about exons and introns. The exon is actually the coding region, and we coined that term because of the expressed region. That's where exon comes from. And then conversely, you have the intron, which is a non-coding region. And then there's also some other parts of a gene that aren't necessarily translated, and they really help more from a processing standpoint. Not only are those the introns, but there's promoters and splice sites and other kind of regulatory sequences. This is one of those things, as we've done genetic testing, that has really changed with time, where we know that actually, if you have changes in those areas, even though they're untranslated, that can still affect your protein product and how things are functioning in the body. And so that's one of those ways we have expanded our understanding of genetic disease in general. a few additional terms. So we'll talk about chromosomes, and I think about that as being the way our DNA is organized. So that's that tightly wound structure, and these we can see under the microscope with the correct procedure to process a blood sample. The genome is the entire DNA sequence for a person. So that's all of the genetic information. It includes that coding region and everything else, whereas the exome is just the coding region, as in just the exons. And those are going to be really important as we're talking about testing going further too. Sometimes I think it's hard to talk about DNA with families because it's something that is so abstract, and it's not necessarily something tangible, and that can be a struggle when you're counseling for testing and things. So going back to the basics is often really helpful for our families, and I like this slide for that reason as to why do we care about genes and how do they make up the body. And that's why I threw this in here, because then it just kind of puts the bigger picture together where, gosh, if you've got something wrong with the body's instructions within the cell, those cells don't work right, then therefore the tissues may not form quite right or work right, and so on and so forth up to the organs and things. So that is just kind of our review in general of some of the basics of testing. Because we use all those terms, I like for you guys to have them all mapped out. But really what we want to talk about is testing. So when we're doing genetic testing, our goal is to evaluate the DNA in some form or fashion so that we can find an answer for a constellation of symptoms. There's lots of different genetic tests, and that kind of depends on what you're looking for and for different conditions. Specimen requirements are also going to vary by test too, and so making sure you get the right tube of blood is important for whatever test you're going for. At this point, there's not a readily available test to check for everything. This is somewhat changing. We're going to talk about newer technologies, specifically for some things that we're doing at Children's Mercy, but that depending on your institution, you may not have access for just yet, but things coming down the horizon. Now, when we think about genetic testing, a lot of times there's just a couple of different categories, and it's a kind of a framework in thinking about not only how you counsel a family, but also in what you're looking for. So cytogenetic testing is really looking at chromosomes. So that's things like FISH and karyotype, which we're going to go through in a little bit more detail. I also think about this as dosage or amount. Do we have the right amount of genetic information? And so if you think about DNA as being instructions, if you've got an extra paragraph of instructions, your body may not read that quite right, and that can cause a problem. Or if you're missing a whole paragraph, I mean, think about it like an IKEA bookshelf, right? If you're missing a whole paragraph of instructions, we are in big trouble for what's going to happen to that bookshelf. And so I think about this as what is our amount of genetic information. And for you guys in allergy immunology, I think the most common thing where that is a consideration is like in 22q deletion syndrome. What happens there is we're missing a part of the long arm of the 22nd chromosome, and there's multiple genes in there, and that causes a whole constellation of symptoms based on that difference in dosage. The other side of things is really the molecular testing, and this is more DNA-based analysis, and I think about this as zooming into the individual letters. So this is sequencing or spell checking. Is there a spelling difference in the way that the DNA is read? And so this is for a lot of your like single gene immunodeficiencies. I put in adenosine deaminase deficiency because I think it's just one that we all think about and know about. And so if you're concerned about that condition, you're going to look at that gene and you're going to spell check that gene to try to find the spelling error, because that condition is mostly caused by sequence changes. And that's how we would think about those sorts of things. Sometimes I think putting it into these categories is also really helpful for families. It's kind of like we're checking maybe quantity and we're checking maybe quality, or we need to look at both depending on what you're worried about. And that's something that I think a family, though they may not get the nuances of DNA and it's hard to understand that, but they can understand absolutely like, okay, this is how we're going to make sure that my kiddo has the correct instructions for what's going on. Or if we don't have those correct instruction, what does that mean for future and how do we manage things going forward? So all of these things, though it's really important to understand what you're doing, they also have basis in counseling too. Now, I recognize that in allergy immunology, you're not ordering a ton of cytogenetic testing. Totally fine. I like you to know, though, where we came from. And I think especially for some of the older kids that you see, you will see kids that have had some of this testing. So just to be familiar with what's happening and what that testing looked for, what it can't look for, and then looking at those what do those reports mean? So we do have our karyotype. That is our actual pictures of chromosomes. And really, in the early 2000s, this is what we had. We had karyotype and FISH, and these were really our main things that we could do. We've had karyotype actually since 1959. And what we do is get a sample of peripheral blood, and it's the T lymphocytes that give us the DNA. We culture that sample. We arrest the cells in metaphase. I know, back to the cell cycle, but that's when the chromosomes are really, really dense. And then we can actually release the chromosomes from the cell and fix them to give us these pictures. And based on the banding pattern, we can look at the arrangement of that genetic information. So we see things like large deletions, large extra pieces, a piece that's inserted somewhere it's not supposed to be, or a translocation where there's pieces that are moved around. The problem is that we can't see things that are really small, and we certainly can't see down to the letter. And so it is a pretty limited test. I will say, I think for you guys in general in clinic, you might see kids with chromosome differences that had this test. So they may not have like a primary immunodeficiency, but as part of their chromosome difference, that may be part of the manifestation. So that's why I still bring this up. I think it still comes up in clinical practice just when you're thinking about some of your kiddos. Now, FISH is more targeted than a karyotype. What you're doing here is you're labeling these DNA probes, and they light up in their fluorescent. We got a couple of pictures here as to how they light up, and you're basically counting to see is there extra or missing. And so for a duplication, you would see 3 probes light up as opposed to 2, and for a deletion, you would only see 1 probe light up. This is a very specific test, right? Because you have to pick your probe, and so you have to have clinical suspicion. So if you're not quite sure what's going on, FISH may not be your best test. And for many, many years, these are, these are really the only 2 options that we had available. And I should say, when you look at results, a lot of times what you're going to see is just the actual nomenclature. So 46XX or 46XY is normal. That's either male or a female. You can see kiddos with trisomy 21, for instance. There's that extra chromosome, which is reflected in the number and then telling you what the extra chromosome is. There's lots more interesting things about nomenclature, and the coordinates, of course, are important too. The bottom line for you guys, though, is just when you're evaluating kids who might have had this testing done, what is that report telling me? And it's just giving you the amount of DNA, the sex chromosomes, and whether there's these rearrangements that are present or not, and specific coordinates for said rearrangement. So I think about this like GPS coordinates a lot of times. It's really just localizing you to a specific area. For FISH, this is the clinical scenario that I think about you guys for FISH. If you've got a kiddo who has a heart condition, a congenital heart defect, and you've got a cleft palate and hypocalcemia, your first thought might be, ooh, we should order FISH because this sounds an awful lot like 22q. And that is not a bad thought, and for years and years, that is what we did. The problem with FISH for 22q is that fluorescent probe is not a one-size-fits-all, and so it can miss smaller deletions if you use a probe that is too large. And depending on your lab, which you may or may not have control over, you might have a pretty big probe that it won't pick up everything. And so that's really important. If you get a FISH back and it's negative, and you're like, well, gosh, I thought that would have checked that. You could still very well be right, and it could still be 22q. It just means that maybe the probe wasn't quite right. So that is something that we think about. This is very institution dependent, I find, as to whether or not people recommend FISH for 22q. I can tell you that I do not recommend FISH for 22q typically. I also like having the microarray result to tell me how big that deletion is and to give me a little bit more parameters. It's not necessarily that management will be a ton different, but it is helpful in that regard. And then I'm not, if my FISH is negative and then I still go to the microarray, it's taken more time and I've spent more money. And so it's not even always the most cost-effective thing to start with FISH first. There's also other options to detect 22q. We will talk about those, but just so you know, generally speaking, that's kind of what we're thinking about when we're seeing a baby in the NICU when there's concern for 22q. We're likely not going to jump to FISH first rattle out of the box. Now, for years, those modalities were really what we had for genetic testing. And we don't order those things really as much anymore because genetic testing has expanded and we've gotten more efficient and we can look at more things. And that's really important. In 2004, the chromosome microarray became clinically available. And this was really big in the land of genetics. So this is still a cytogenetic test, meaning I'm looking at the chromosomes and I'm looking for missing or extra pieces, but my goodness, I can see some smaller missing and extra pieces, which is really important. There are different types of array. You will see maybe on old reports of older kids a CGH array. We don't really do those anymore. We tend to use what's called a SNP array. And when I say SNP, it's SNP in all caps. And that's an abbreviation that stands for single nucleotide polymorphism. And all that is that there's a difference of a single nucleotide, literally one base pair at the same position in genomic DNA sequence. And these are things that are common. These have a frequency greater than 1%, and that's what a SNP is. So we have a bunch of SNPs all throughout our genetic information. And that's what we use to kind of steer us towards these missing or extra pieces and to kind of figure out what our amount of DNA is. The microarray has a gene chip, and there's a large number of these little DNA sequences, these little SNPs that are immobilized on that chip. We pop the patient's DNA sample in with the chip so that it can undergo hybridization with the probes on the chip, and then a laser actually goes through and scans that chip, and we can look at the dosage based on the signal intensities, which is kind of cool. So there's kind of this schematic of what you might get out, but we end up with kind of these graphs. And so as you can see, you might have a more intense signal for something that's a duplication because there's more of it. There's 3 copies instead of 2. You can see your 2 copies kind of treading right in the middle, and then a deletion where there is 1 copy. The benefit of using SNPs is that there are so many that are identified, so you have so many points to look at, which means you can check really small missing and extra pieces, much smaller than we're ever going to be able to see with a karyotype. So that is really helpful too. Even thinking about some of our single gene conditions, sometimes we can see down at the exon level, and so we can figure out somebody might have a small deletion that makes them a carrier for an autosomal recessive condition or things like that. So this was really a big deal in testing and has continued to kind of improve with time too as we keep identifying SNPs. This is processed a little bit more like a DNA test, even though it's a cytogenetic test. So here's what I mean by that. For the previous test, we're talking about like culturing cells and doing those sorts of things with them, right? We have to get the cell conditions right to even be able to perform those tests. Those test a karyotype and a FISH actually go in a sodium heparin tube. That's a green tube for that reason, because of how we have to make sure that the cells are ready to be processed. In DNA testing, we use a purple top tube or an EDTA tube. And so with a microarray, you use that type of tube. Even though you're looking at a chromosome condition, this is still, we don't have to culture the cells for a microarray, and that's one of the differences as well there. Now, what a microarray can't do, it can't get down to the individual base pair level, and it can't tell me how the DNA is arranged. So even for some of our kids that get a diagnosis for microarray, I still got to backtrack and do a karyotype so that I have the arrangement correctly, just from a recurrence risk standpoint. Not something that you necessarily need on your side of things, but that's why for a lot of our kids, you might see both of those tests in the chart. what do these results look like? So with a microarray, the ARR stands for array. That's just the type of technology. And then as we saw earlier, everybody has, humans have 46 chromosomes. They're divided into pairs, and they're numbered 1 through 22, and then we have our sex chromosomes. For females, that's XX. For males, that is XY. And so this is a normal female because there's 2 of everything, and that's what we would expect. Everybody gets a copy of genetic information from mom, and then they get a copy from dad too, and that gives us our full, complete genetic set of information. For a male, you have your 1 through 22 with 2 copies. Makes sense, right? Same kind of thing. But you only have 1 copy of X and Y. That's a normal male or a typical male. And that makes sense because that is, it's not that there's something missing. It's just we expect there to be that sex chromosome difference. Now, if you have something that is causing a problem, or that is a pathologic diagnosis, right? That is where some of this nomenclature comes into play. We still use coordinates like we would with a karyotype, but we also have some additional information like these numbers that can give us more specific at where the breakpoints are for this chromosome change. So for instance, we see the array. That is the technology that we've used. We're on chromosome 4. Q is the long arm, and then these are our coordinates, and it goes to the end of the chromosome. And that times 3 tells you the dosage. So this is a duplication at chromosome 4Q from 28.3 to the end of the chromosome. Now, I am not expecting you to know all of that and to remember all of that because there's 9 million examples we could go through, and there's all kinds of nuances with this. But this is what that test report would look like. Now, this is a different result, clearly. So I gave you one also that has a deletion, so you can see. So again, it's an array on chromosome 17. This is on the short arm. That's the petite arm, or P, at coordinates 1.2. And then these are our specific breakpoints, and that's times 1. That tells me there is a deletion there. And then the report will help clarify. Remember, depending on where you are, if you have, I hope you have access to friendly neighborhood geneticists and genetic counselors that can always help you with this, but this is kind of the quick and dirty of what you might be looking for with this test report. So that's it for cytogenetic testing, because that's not really for you guys where you would necessarily focus. So I'd like to talk a little bit more about molecular testing, and that is more and more what we're coming out with and where technology is heading. So this is where we're really talking about sequence or spelling changes, and we're really talking about one gene. We know, as we have done a lot more genetic testing, that sometimes kids can have more than one thing going on, and so there's some complexities in that. But when we're looking at a clinical picture, we're really trying to put the features together in one genetic condition to offer that explanation to the family. For many of our conditions, the disease-causing sequence changes are going to occur in the coding region, and that is a very traditional sort of way of thinking, because we recognize that that coding sequence is so important for the protein structure. I said disease-causing sequence changes here. A lot of times in the past, we would use the term mutation. I think mutation is actually a really helpful term because it's implying that it's causing a problem, but some people really do not like that term because it could also imply that the person with the genetic disease is a mutant. I do not think of it that way, but I recognize being sensitive. Most of the genetic literature these days and in conferences and how we'll talk about things, we'll talk about pathogenic variants. I find that to be a bit of a mouthful. But it is just kind of where things have gone. So if you see that in a paper or in a report, that's equivalent to mutation. And really, we're talking about genetic changes that cause a problem. The other thing to think about too is, as time has gone on, we also know that other parts of the gene can be really important when trying to identify a genetic disease. We find changes in the introns now that cause problems. We find changes in the promoter region that actually cause disease. And so the coding region is not necessarily the be-all end-all, but that is where we started in kind of getting genetic testing. And the picture of the origami is just really helpful for me to remember. If you have an instruction that's messed up, you're not going to get the crane. You might end up with a crumpled ball that doesn't tell you what things were supposed to look like. And that's really what we're going for when we are doing genetic testing. We're trying to figure out why things aren't working or aren't formed the way that they should. So from a sequencing standpoint, what we're going to primarily be talking about is next generation sequencing. This became available in 2012 clinically, and this was really the next big advent that came about so that we could diagnose single gene disorders. And there's more than one type of sequencing test, so sometimes you might just be looking at literally one gene, and you're looking to see, is there a spelling difference? I love this slide because I think it's so helpful to think about, as we said, DNA is abstract and it's kind of hard, but this gives you like a visual of like, oh, I can't make out that the car was actually red now because of the spelling change, so therefore that same meaning is not communicated. A lot of times, especially as we learned about multiple genetic causes for a clinical picture. So for you guys, I would say immunodeficiencies is a great example. We would use targeted panels. So somebody at a lab, whether it was an academic lab like we have at Children's Mercy or a commercial lab that you might use, would go through and curate that panel and say, well, we know all of these genes cause problems with the immune system, so if somebody's worried about that, they can order that test. And you can see different sentences there. We're looking at different things, and the bottom line is for each of these sentences, we're trying to figure out if there's a spelling change in any of those sentences that would cause problems. And just an example of what that could be like when you're explaining to a family. We also have exome and genome sequencing. So with exome, since we're only looking at the coding regions, that's looking at like the highlights, the most important chapters of the book. Whereas genome, you're looking at everything, right? Like sometimes when we read, we might omit the word, like some of the adjectives like the and a, like those sorts of things. Genome looks at everything. It's going to go from the very beginning to all the way to the very end, and there's not skippage into that. And so that's where you get kind of that breadth of testing. Now, before we had next generation sequencing, really the sequencing that we used was Sanger, which we all probably learned about in college biochemistry, right? Where you're like making your little primers and you're trying to, and you're doing this for a single gene. And it was really laborsome, and trying to do that on a large scale for all of these genes was really a struggle. Sanger is great, but you got to know what you're looking for so you can target that gene. And because it takes a lot of work, it's also pretty expensive and it's going to take time. So in order to do much more sequencing on a bigger level, we need a new technology, and that's where next generation sequencing comes in. I will abbreviate that NGS just so you are aware, but that is just the name of the technology. It really came through and streamlined this process. And this is massively parallel sequencing that provides high throughput data from multiple samples at once. And this is therefore a reduced cost compared to Sanger. So let's dive into that a little bit more. What are we doing with NGS? We can simultaneously sequence millions of small fragments of DNA. So in our workflow, we got to get the DNA extracted, right? So we've got that sample ready to go. And all of this, we got to prep the library with all of the little DNA polymerases, adding these fluorescent nucleotides. And basically we're making all of these growing DNA template strands. And the nucleotides are incorporated, have this fluorescent tag for which they're identified by. Bottom line for this is that we can look at millions of fragments simultaneously per run, where Sanger can only sequence a single DNA fragment at a time. So thinking about that, that's why it's so much faster and you have the ability to do more. In this workflow, as I said, we started with our extraction. Prepping the library is just getting all of your fluorescent nucleotides and everything ready. We literally go through, there is a sequencer, that is a type of machinery that goes through and gives you that spelling sequence. And then there's these computer programs that essentially spit out these files and will help you identify, are there spelling changes that are causing problems? This is also more power to detect novel variants or rare variants, and even go deeper into sequencing too. So there's lots of benefits to this technology. We kind of went through this before with our images of thinking about the book. But just so you know, there's different ways of ordering these tests. Site specific is when you're testing for a specific known pathogenic variant. And a lot of times this is like known familial member testing. Say you have a child in clinic that you've diagnosed with ADA deficiency and you know both of their pathogenic variants, right? It's autosomal recessive, so you got to have two. And then the parents come to clinic with their kiddo the next time and they've got a new baby, and they're worried about the baby being tested. Well, if you're worried about ADA deficiency, we've narrowed down to one gene, and we already know what changes run in their family. So it doesn't really make sense to go through the whole gene. You can just specifically look for those changes. And that's kind of your site-specific stuff. Going down through the list, if you are really confident that you just need one single gene, you can just look at one gene, and that is okay. In allergy immunology, I don't know how much that happens for you guys because I feel like there's a lot of fluidity at what you can see in clinical presentations. So sometimes we tend to go broader. That's been my experience. Please correct me if I'm wrong. But I know, for instance, in genetics clinic, something that I think about doing this for is like neurofibromatosis type 1. If somebody has multiple cafe au lait spots and neurofibromas and freckles under their arms, I need to just look for NF1 because those are hallmark features. And there's not anything else that's going to give me all three of those things. So that's what I'm going to order. I don't need to test for other things because that's not what we're looking for. Specific disorder panels, I think that's probably something that in the past you guys have really done a lot of with like an immunodeficiency panel. So really looking at a group of genes that will cause some a similar clinical picture or the same genetic disorder. That's really what we're going for there. Now, moving on to bigger testing, whole exome sequencing. This is probably the largest test that's available at many centers, and that I would also say is probably the easiest larger test to get approved by insurance. Insurance is always going to be that thing in the background that might limit us a little bit on genetic testing. So things to think about. There's approximately 180,000 exons in the human genome, and that's about 3% of the genome. That is not very much, right? But it's still a ton of information, and that's why the turnaround time tends to be longer than maybe some of your panels. Panels are often 3 weeks, 4 weeks. Exomes tend to be more 6 to 8 weeks at that point. Other things to think about with exome testing. Since you're looking at more genetic information, we are going to find changes. And that's why we recommend doing trio testing or getting samples from mom and dad too. It tells us what is normal for that family. It tells us if something is brand new in your patient, and it tells us if something is inherited, like parents or carriers. And that can be really helpful in trying to figure out what changes am I worried about on this report. We all have genetic changes, and it's okay. Those things make us who we are, and we don't want to erroneously assign something that's not causing problems to a diagnosis and then maybe even result in management changes that aren't appropriate. So that's one of the reasons we like trio testing, just because we're looking at so much of this information. The other thing to think about too with exome is that because you have all of the sequencing data, you have the opportunity to find things that you wouldn't be looking for with a panel or with any other kind of more narrow test. So there's in genetics, we talk about secondary findings. The American College of Medical Genetics and Genomics actually has a consortium panel that creates a list of secondary findings. There's currently 81. I think when I started, we were like at 50, which it's been a hot second. So we've added some conditions, and families can choose whether or not they want to receive these findings or not, and there's a consent process. These 81 genetic conditions are often often related to cancer predisposition syndromes or cardiovascular syndromes. So for instance, BRCA1, that's a secondary finding gene. If I were to find that incidentally, I would have something to do about it. If it's from mom, we got to do different types of breast cancer screening, different modalities of imaging, and she needs to have more high-risk level screening. On the flip side, from a cardiovascular standpoint, lots of our arrhythmia disorders where we do screening or prophylactic beta blockers or things like that are on this list too, because these are genetic changes where there's management recommendation. There are not things on this list where we're going to be like, sorry, you got this, there's nothing I can do about it. They all have associated management changes. However, families have to consent for those things and opt in or opt out. Some people just don't want to know, and that's okay. And sometimes it's also related to what's going on with the kiddo at the time. If they're really stressed and their kiddo is really sick in the ICU, may not be the best time for thinking about, like, am I going to have cancer one day? I get that. And so we can be dynamic. Many labs have a consent, particularly lots of the commercial labs that you may use occasionally for their exome sequencing. And these things apply to genome sequencing too. This is all your genetic information, so you get your exons and your introns, and so you can get this same additional information of like secondary findings and things like that. Okay, now moving on to why exome versus genome. I really like this picture of the apple because I think it's so helpful in conceptualizing what you're getting versus each test. A genome is no doubt more comprehensive, but it's also harder to interpret because it makes it harder for us to understand the genetic changes in the non-coding regions of DNA. We don't always know if a change in an intron is going to cause a problem, or if it's just going to be like, oh, just a little typo that we read through and no big deal. That makes it really hard to interpret, and it can increase some of those uncertain results. You know, about, I don't know, maybe 10-ish years ago, we really thought that the genome was going to increase our diagnostic yield a ton. And it's not that it doesn't. It does a little bit, but there's still a lot that we're learning. And a genome can't pick up on every single type of genetic change, too. It's really good for the spelling differences in that whole sequence, but there's other things that it's not as good at picking up on. Of note, the genome is also pretty good for copy number. So when we talk about copy number variation, or CMV is how you're going to actually see it abbreviated a lot of times, that's an amount kind of question, but those tend to be smaller missing or extra pieces. So when I think about that, I'm thinking about smaller deletions or duplications within a gene that you might not be able to pick up with microarray. Picking up those missing or extra pieces wasn't as good with the original exome technology. And so that was something that we missed. And that's why on so many kids' charts that you would read who had seen genetics, we'd start with a microarray. And then if that was negative, we would go to exome, because we're getting both our quantity of DNA and our quality of DNA. And we couldn't overlap both of the tests. Now this is changing, and we're going to talk about that, but just so you know, that was some of the method to that madness. Now, the other thing to think about too, as much as our goal is really to do what is right for the patient and to get the most appropriate test, insurance and cost is always going to be a barrier. And many insurance plans do not want to pay for genome sequencing. A lot of times they want to pay for genetic testing in general, and they'll put up all kinds of barriers in place for that. Genome is absolutely more expensive because you're doing, there's more information to test, and depending on from the wet lab standpoint, some of the reagents and things are different too, to be able to look at that specific data. So there's definitely a cost factor that comes into this. So these sound great, but they're not perfect. So what are we missing? Now, if you do an exome, you're only getting the data on what you're sequencing. You're only getting codons for the exons, right? You're not getting the introns. Are you missing something in a promoter? Hard to say. We didn't look at that. Panel testing. This is one that I think is interesting, and something that we have in genetics. I have seen this happen over the years, and it's kind of changed the way that I order genetic testing personally. So a lot of times I really try to hone in on what I think is going on with the kiddo. It's helpful for like the team that's consulting me. It's helpful for the family. And if I can narrow down my testing and maybe it's more cost effective, that's great. But sometimes that test comes back negative, and it's because I didn't look at the right thing, right? I didn't order the right type of panel, or maybe the clinical picture wasn't a great fit for what the kiddo actually has. You know, our kids do not read the textbook. If I have learned anything, it is that. And so sometimes being broader when you think you could go into more than one category is really helpful. I think for some of the patients that might you would consider an immunodeficiency panel, is it possibility that maybe they could have a primary ciliary dyskinesia, and that's why they're having these frequent infections? Depending on your panel, you might not get those genes, so your immunodeficiency panel may come back normal, and yet we're missing what's actually going on with the kiddo, and then how we need to manage it. My kind of rule of thumb is that if you're considering more than one panel, I would probably just go on ahead and get at least an exome and call it a day, so that you get the broad evaluation. And ultimately, I know from a cost standpoint, we make the argument a lot. Actually, it's more efficient and it's more effective to just do the big test and be done, as opposed to cherry picking different tests, because it will delay your workup. There's multiple publications about the cost of additional unnecessary evaluations, sometimes even harmful things that happen in trying to figure out what's going on with our kids. So really important to think about and be mindful that, sometimes going big, it might be the best thing. Now, historically, as I said, that those exomes didn't pick up some of the larger deletions. This has changed. Technology has improved. We're going to talk about that more, but this has been a historic limitation, and that is why we would always also get a microarray. Sometimes you're spell checking your sentence, but it doesn't tell you whether or not your sentence actually makes sense. I think about that in context of different types of genetic changes. I don't harp on those a lot for this presentation, simply because they're not things that you guys see quite so much in allergy immunology. But these would be things like repeat disorders or methylation disorders, right? Where they're different types of genetic alterations, and that spell checker doesn't do a good job of recognizing those things, so you have to do different testing. The other thing that's a problem is that sometimes the spell checker may not recognize it and say, ooh, this may be a problem, but it doesn't mean that it's causing a problem. That's what I'm talking about with these changes that we all have in our DNA. These changes make us who we are, and they don't all cause a problem, and we really want to hone in on what's giving our kiddos the symptoms that they're having. The other thing too is that, we're still learning. I know, especially you guys, I feel like the immunology literature has absolutely blossomed with all of these new conditions, and we don't totally understand the full clinical picture and all of the manifestations. And that can be a struggle too when we're trying to do testing and going, could this fit? Is this right or is this not right? And how do we figure that out? So when you get results back from genetic testing from sequencing, these results are going to be described in two ways. The first is at the DNA level, and the second is at the protein level. And so at the DNA level, you will hear us refer to that as the C dot, and it'll give you a position that is the 83, and then it'll tell you the G to A. That means that there should be a G at that position, and it's been changed to an A. That's the coding reference sequence, and that's where we see the change. Now, at the protein level, we'll call that the P dot, and that'll tell us, well, this is the amino acid that's supposed to be here, and at this position, it's actually this amino acid. So we've got an example here, that P dot VAL 312 ALA, and then I also put it, because sometimes some labs will do the amino acid abbreviations with only the one letter. I literally keep a thing at my desk that has them all, because sometimes I forget and want to make sure I've got my correct amino acids in my head. Anyway, this means that the valine that should be in the 312th amino acid position has actually been changed to an alanine. And so those are the two ways you'll look at things. When you're looking at papers, sometimes they're inconsistent and only giving you one or the other, and it's really helpful when you're trying to get more information about specific variants to have both of those factors. So that's why I wanted to break down what those are. Types of genetic changes. So sometimes we have missense changes. Those just change the amino acid. And you can see what that would look like in our sentences that we were using earlier. Instead of saying the car was red, we have the car was hat, or the car was RDD. And so does a missense mutation always cause a problem? With the connotation of mutation? Absolutely, we can see genetic disease from these, but there can also be benign variation too. That makes it challenging to interpret. We can also see nonsense mutations. This is where we get a truncated protein, and then a frame shift, which is where there's been like a base that's been added or something that's been deleted, and then my whole reading frame gets thrown off. So for a truncated protein, you'll see an X as the second amino acid, or sometimes you'll see a stop. I don't see stop as much anymore. I think the literature has changed to really do the X, but it is something that you might see in older reports. And then from the frame shift standpoint, I don't even know what's going on in those sentences, right? Like, we're totally off kilter. When we started doing genetic testing, this is really what we were looking for, right? Because you're going, how could you possibly make a correct protein with that? And now we know there's a lot more nuances, and there's some genetic conditions that are caused by only one type of variant, and some are where you get a different phenotype with another type. So there's a whole host of things that have blossomed just from this type of variant, but this is just to give you a little bit of a foundation when you're thinking about the genetic changes that you're seeing in a report. So from a test result standpoint, of course, the goal in doing testing is to understand what's going on and get a positive result. So that means you found something that was likely pathogenic or pathogenic, that's detected, and it fits with the patient's clinical features. That fits with the patient's clinical features, that's kind of up to your clinician. There are times where sometimes the lab is like, we found this and we think it's great. And I'm like, it doesn't fit with my kid. So either we have something new going on, or something is incidental, or there's something not quite right. So just so you know, that clinical feature part is really important. On the flip side, you also have a negative test. So no variants were found, or only benign or likely benign variants were detected. We don't put those on the chart, on reports anymore. Generally, we all have a ton of benign variation. A lot of times, if they're on there, it's because they were initially classified as a variant of uncertain significance, and it's kind of the way to close the loop. We no longer are worried that this could cause a problem. This has been reclassified. Now, the variant of uncertain significance often is the bee in everybody's bonnet, and that is where we found a change in a gene, might be related to the patient's clinical features, but we're not sure if that change actually causes problem, and we need more research. Those are a challenge, and it's not going to get better as genetic testing evolves, because we're learning new types of tests and we're able to detect more things that we don't understand. So just be prepared when you're doing testing. It's just not all like very cut and dry. There's lots of nuance in this. And so we tend to warn families when we're doing testing too, that like sometimes we get uncertain results, and for families with a lot of anxiety, that can be really hard. So just to give that heads up too, that like we're going to walk you through this. Most of the time we don't change what we're doing based on a variant of uncertain significance, because most of the time those end up being reclassified as benign. But there's other circumstances where they're a little bit more concerning. And so there's nuances in that. And as you do more and more genetic testing, I think a lot of times you get a little bit of a better handle as to what you're going to do with that VUS. But most of the time we actually don't change much based on it. I like this scale too, because I think it's helpful when you're thinking about these genetic changes and like, how worried should I be? So as we've been doing genetic tests for a long time now, or longer time, I should say, a lot of the VUSs that we initially found ended up being benign more than 90% of the time. That is so helpful to have that statistic in the back of your mind, and to kind of know a lot of the things that you see that you're like, we don't know what to do with, down the road they may actually be reclassified. And then of course, pathogenic and likely pathogenic are going to be your diagnostic results that are coming back. So this is not something that's going away. We're going to see more uncertain results as testing broadens, but just for you to be aware about as you go through and order genetic testing and you see some of these results come back, how do you think about those things? Now, I will say, again, kind of going back to basics, but I think that the patterns of inheritance are really important when you're looking at a test report. Inheritance is key for making the correct diagnosis. It's also important in preventing an incorrect diagnosis. So understanding if you have one pathogenic variant in a recessive condition, it's going to say that that change is pathogenic, and it is, but you need another change to cause disease. That's really important, and sometimes people forget that, and sometimes parents need to go through that again and need that explained to them. The other thing that I think inheritance is really key for is like, who else needs testing in the family? Because, you know, when you're doing genetic testing and thinking about genetics in general, rarely are you dealing in isolation with just one person. You're really thinking about the whole family. So even if that's not where you're geared toward, you might end up thinking, I may should send these people to genetics so that we can get the rest of the stuff lined up and make sure I'm not missing anything, or see if there's anything else that we need to do there for the rest of the family. Reports are going to tell you about inheritance associated with that gene, and sometimes you will see that there's more than one type of inheritance. So a lot of times I'll see AR for autosomal recessive / AD for autosomal dominant. And then I got to go look at the specific change. I got to look at the patient's clinical picture. I got to look at what's reported with both things. Fortunately, my hope is that you have your friendly neighborhood geneticist or genetic counselor if you need some assistance, because these things, you often have to dive into the literature and into some of the databases, and they do take some time too. And sometimes it's a little bit more up in the air, where one clinical picture has been really well defined, but maybe your kid doesn't fit so well in that. So there can be some nuances to this too. Quick review. We've got our autosomal dominant. We know that this is going through the family. Males and females are equally affected, and the chance of passing on the trait to boys and girls is 50%. And so this is the one that's filtering through all the generations of the family. I recognize that there are other genetics concepts that skew what this looks like. Things like variable expressivity, like yes, they have the condition, but they're not as severe, or they have different clinical features, or incomplete penetrance, which has really become more of a problem these days. And what happens there is that, like, somebody may have the genetic change, but they just don't show symptoms. And that really can skew what your pedigree looks like. I'm not going to get into a ton of those things, but it's something to keep in mind that may make interpretation of results a little bit complex. Autosomal recessive are when parents are carriers and the affected individuals are their children. So parents don't have symptoms typically as carriers. There are clearly lots of exceptions to that rule, but that's how we think about them. Males and females both affected, and the recurrence risk is estimated to be 25%, and I would note 25% for each pregnancy. One time we had a family who said, oh, well, we had our affected child, so we didn't think we would have any more for the next 3. It doesn't work that way, and it's something to think about as you're counseling, and you think, I've been as clear as possible. Maybe not. So just making sure you're keeping those things in mind too. Autosomal, a lot of y'all's like rare immunodeficiencies that are pretty severe are autosomal recessive, and so it's important for those future kiddos and figuring out parents' carrier status, and that can be helpful in making that diagnosis too. And then of course X-linked, where we see things that are really exclusive in males, or they could be more severe in males, or even more common in males. And you're, from the standpoint of depending on who you're looking at in the pedigree, that's going to determine your recurrence risk. I'm not going to go through all that a ton, but just know you will have these slides, and if you have questions, let me know. Now, at Children's Mercy, I just am going to talk about what we do here a little bit, and some of this does apply to outside labs too, from a commercial standpoint. So oftentimes when you're thinking you want to order broad genetic testing and you go to type in what you're going to get, you end up with a symptom-driven whole exome sequencing with copy number variant analysis. What does that mean? Well, we're spell checking through the protein coding parts of the gene and known disease-associated genes, and then we're getting copy number variant. That actually replaces the microarray, which is really cool because I'm getting both tests in one. And I love that for time, I love that for cost, and I love that for how much blood I need, right? We like to put everything together. So this is a really nice test. Other clinical labs are doing this now where they're including copy number variant. For our lab, we are still guiding our analysis by symptoms. That's why we say symptom driven, but we'll still take off filters to look for things that are very obvious and to make sure we're not missing something that kind of came out of left field. We do occasionally find some incidental findings, but if it's something where it would affect health or development, the lab lets us know. And we do recommend parent samples. We can get this back in 4 weeks for stat. That's typically inpatient ordered, but you can do that for outpatient too. You skip insurance prior auth, and so there could be a cost issue for families. 12 weeks is our non-stat turnaround time once we have insurance approval, and this is the most common thing we order. I will say for us at this is exclusively through Children's Mercy Lab, but our symptom-driven exome sequencing with copy number variant backbone or testing platform actually is a genome. So you're getting really, really good coverage. So it's kind of like an exome plus is how I think about it. And this is one of the tests that we recommend frequently, and that is ordered a ton of the time throughout our hospital. And truthfully, for our exome here, the genome, I rarely order because I think our exome does such a good job of what we're doing. Because it's symptom driven as well, we don't have a consent form. You guys, I'm sure, know that because you're not getting parental consent in a written format. And the parents don't have the option to receive secondary findings. We just don't even look for them. If something flies off as being, oh my gosh, I know this change and I know it's associated with this, the lab will tell us. And so that we can make recommendations going forward if that's something we need to do. So how often do we get an answer? It depends on what you're looking at, and it depends on your indication. So range typically has a positive yield between 35 and 45%. The phenotype is also going to contribute. So what's the clinical picture you're dealing with? So like hypotonia, great yield actually. Multiple congenital anomalies, that's something that has a pretty decent yield. That's why we started doing this kind of testing in the first place. Things that are newer and we're still learning about, interstitial lung disease, I think is a great example. Lots of times, we're checking to make sure it's not that, but the yield and actually find a positive is low. And I think it's really frustrating for lots of people that we consult for and that we work with, that like we may not get an answer, and we get frustrated with that too. And it can be a struggle when you're going, I know something is going on here. And non-diagnostic test results can be helpful too, because then you're ruling out some things. Now, I don't know how aware you are of this, but right now, Children's Mercy has a unique opportunity. We have what's called PacBio Hi-Fi sequencing. This is something called long-ring sequencing, and this is different than our next generation sequencing. And the thought is that this is really going to be an all-in-one test. You get your genome, right? So all that sequencing we talked about, you get your copy number analysis. So that's your microarray, but you also get things like methylation and repeat numbers. Really, really cool, right? In one big test. This was initially available through research, and now we are doing it clinically on the inpatient side of things. Most centers do not currently have access to this technology, and our lab is cranking these out in 4 weeks, and it's been really interesting to see some of the results and stuff that has come back. So if this is something that is available, a lot of the kids in the NICU and the PICU actually end up getting Hi-Fi at this point, and the lab kind of has a process to see who's a candidate for that and if there's anything additional that we need to do. So moving on from testing, I just want to fly through a couple of things for allergy immunology and what's kind of new in the land of genetic testing for you guys. Back in the day, I really feel like there were all of these papers that would have these massive algorithms about check this immune cell population and then do this and then do this. And then once you've done 9 million labs, you're going to genetic testing as kind of the confirmation, and you're really trying to hone in before genetic testing on what's going on. And I think that's really shifting for you guys. Not all of our kids read the textbook, as we talked about, and sometimes, you get labs at a point and they're normal, and then you get them down the road and then they're not normal. And how do you account for those sorts of things? And so a lot of papers are really advocating for broad genetic testing. So this is one that I thought was pretty interesting. This came out in 2021, and it was looking at panel to exome. And what they ended up finding was that there were people that where they certainly found the diagnosis with the panel, and it was helpful, but then for the ones where they didn't find it, they went on to do exome and they found the diagnosis. And what this paper was advocating for was that really makes sense to start with an exome, because it's harder to predict who's going to fit very well in that box. And a lot of the patients that we started it with the exome with were ones that had less common clinical and laboratory phenotypes, so you weren't quite sure what box to put them in anyway, which made a diagnosis a little bit more unlikely. And looking at the ones where we started with the panel, because we felt a little bit more confident, those were going to be things you picked up by the exome anyway. So I think that is super interesting. And they talked about it as being a cost-saving approach too, which I thought was really cool, especially when you're trying to get things paid through insurance. The other thing that I thought was pretty cool was this paper that came out in 2022, and it looked at 1,000 families with complex immune phenotypes and was really trying to show, like, hey, we're going to have to be broad with this. And what I thought was this figure looks at all of these disorders that were found. The blue shading shows variants that had not been previously reported in our databases, which is really helpful to know because maybe they're changes that are harder to pick up or things like that. We also had some copy number things that popped up. Those are designated by the gray. There's dark gray and there's light gray. And so this was a very interesting paper too. And I think one of the things that was interesting about this is that there were lots of new disease gene associations that we didn't know about previously, which means there's a delay in curation for a panel, right? Because we didn't know about the association. I think the other things that was also kind of cool too is that some of these people have more than one thing going on, and that's really important because that's going to skew what your clinical picture is. And so this was also very much advocating that, it actually might be a better idea to start big and go from there. So really, lots of publications are advocating for broader testing to be considered earlier in the presentation, and then maybe using some of your workup that you might have done previously to help from a phenotype standpoint. The other thing that I think is really interesting too is that the phenotypic spectrum of these conditions are really changing. We're seeing so much variability in severity where you have really, really sick kids early on and then people who may not have as many problems. And we're also seeing a lot of variability in some of the lab abnormalities, and I think that's pretty interesting too. And so just because you have a specific pattern or you think you do doesn't mean it's going to correlate to your diagnosis. Now, the allergy side of things we struggle with still, and I think it's because a lot of these things, like your atopy kind of symptoms, are not typically described as genetic. They're often more familial than Mendelian, meaning there's we see them run in families, but there's not a single gene that's going on, or there's multiple factors that come into play, or the genetic things that we've identified are more susceptibility sort of things. So if you have something for susceptibility, that doesn't mean you're going to have it though. And that's the hard part. There are still some things where genetic syndromes exist. I put in peeling skin syndrome. And I think skin findings are something that a lot of times we attribute things to eczema, and then you kind of go digging and you're like, maybe that's actually not eczema. And that might be one way to get you to, do we need to do something different, or could this be something more going on? Again, these things are really rare, but I do think it's interesting to see where we might be going with some of these conditions in the future. I also think just from a severity standpoint, immunodeficiencies are scary, and there are so many management things that need to be done early on. So I think a lot of the research has been more focused in that area, just in kind of as somebody not an allergy immunologist, just as somebody who looks at things from the genetic side of things. And whereas, on the allergy side, we're not talking about doing some of these major things. Not that these sorts of symptoms can't be very severe, because we certainly see them that way. But it'll be interesting to see kind of where the research goes. So on that note and that whirlwind, that is the end of my slides. Do you guys have any questions or any